So I'm Thierry Poumero, the Chief Business Officer of the company. I would like also to thank again Coma Biotech and KG, and looking forward to this partnership and supplying the users in Korea. So today we're going to speak about organ-on-chip. We will talk about the technology first, and then we will move to the applications. And also we have listed a series of questions that we received prior to this event that we will respond to. And of course, there will be a session for question and answer also if you have at the end of the session. So first, to start, we would like just to make some definition about microfluidics and organ-on-chip, which is the technologies and the devices that we have created and designed and produced now. So just for your information, for those who are not so familiar, microfluidics is the science of manipulating some fluids at a very small dimension level, micrometers level, which use some economy of scales and also some capabilities in terms of miniaturization. The organ-on-chip are basically defined as microphysiological systems, or what we call also tissue chips, that are basically containing cells that are cultures, and then the aim here is to basically reproduce or mimicate the various aspects of the body and the human physiology. Basically, the idea is also, due to the miniaturization and using microfluidics, also to be able to use in high throughput those devices to test efficacy or toxicity, or go into drug discovery field. So let's come a little more focus on the compartmentalization and the culture, and then we're going to enter into the technology of NETRI. So basically, generally speaking, let's say until recent years, the standard method for growing cells together was a kind of mixing them together into a single well, which was the let's say, and it's still used as those days. Basically, what we want to do is we want to reproduce the natural compartmentalization of the body, and using basically what the anatomy tells us about central nervous system, peripheral nervous system, and organs. So we will come into that. So focusing more on the devices itself and our technology, you can see here on the right a set of 4 chips, which is our unit of production. And basically, those 4 chips are identical. And then if you go a little on the left, you see again those chips here. So basically, one chip is composed of 6 wells. So on the image, 3 on the top would be the inlets, where you're going to pipette in the cells and the media and whatever biological materials needed for growing the cells. And on the bottom of the image of the chip, you would see 3 wells, which are the outlets where you can pipette out. So the way we design the chips is that we have 3 channels, 1 channel here, a 2nd channel in the middle, and a 3rd channel on the right. So channel 1 on the left, channel 2 in the middle, and channel 3 on the right. As you can see, channel 1 and channel 3 are symmetrical, and the channel 2 is a linear channel, I would say, that acts for separating the fluids and avoiding the fluids to mix together. Thanks to the microfluidics technology, so we have the ability to do co-culture or pre-culture into our chip, and those culture will not see each other. So basically in channel 1, you would get cell type 1 with media type 1, and then it will grow. Separately from channel 3, where you would have a cell type 3 with media type 3 in the channel 3. So basically, this is very important to understand that the culture of the cells in our chip can grow separately. Then, if you go here, you would see, and my mouse is not as reactive as I would like, but you can see here the region of interest where you see on the left blue cells, which corresponds to sensory neurons. And here on the picture, you could see on red type cells, which are keratinocytes, which are cells from the skin. Basically, those cells will grow, as I said earlier, separately. In addition to that, we have created microchannels, that is, you see horizontally on the images. And those microchannels are extremely small. They are about few microns by few microns. And if you remember the explanation about the microfluidics advantages, it gives us the possibility to control the surface tension and diffusion into those microchannels, which means for us here, in those kind of applications, that the neurons on the left will project into the microchannels, so the axons will go into those microtunnels and then will be guided and will go from channel 1 into channel 2, and then from channel 2 into channel 3. Meaning that also, because it's very small microchannels, only the axon can go in, so then there will be a kind of non-possibility of the of liquid to go inside also. So with this technology, we can basically reproduce innervated organs, where on the left you would have reservoirs of neuronal cells, and on the right you would get organ cells, and then you can innervate those organs. That's a key feature, because then you can test compounds, or you can test efficacy or toxicity of any molecules, either on the neuronal part, or if needed on the organ cell part. The other advantage that we have in our technology is we have the ability to let the cells breathe due to the materials that we use, which is a silicon type. We call it PDMS, and it allows the gas exchange. So that means that when you use our devices, you don't need pumps. So it is a standalone device, meaning that it is very easy to use. The other advantage of our technology is, as you could see, there is six wells per chip, and then we have 16 chips into our plate. You see on the black here, black frame. So inside the black frame. So that's the plate, and the plate is designed to be a 96 well plate, meaning that it is SBS compatible format for any lab equipment in the laboratory. So that's another advantage. That means that this plate works as a cell culture device that you can put into an incubator, and then you can also make some analysis during the incubation and after the incubation when you want to test compounds. You can see here some images of the cell growth of the sensory neurons that will basically are on the left, and that will cross those microchannels that you can see on the image between channel 1 and channel 2, and between channel 2 and channel 3. And then you can see the projection of the sensory neuron on the right. So this is a very nice image to show you how good the devices can allow the cell culture. So with this technology, we can, as we said, compartmentalize the different microenvironments, as well as define some isolation of the neurites, which is very interesting for studies on neuronal cells. And then finally, we have the ability to control and isolate the fluids between themselves, so which basically give us also a very good reproducibility of our results. The cell cultures can be allowed, it can be possible in our devices, and as we can see here on this slide, there are some large possibilities of using a different type of cells. And for example, we work with the main cell provider in the world using iPSC cells, which we validated. So you can see some pictures here with glutamatergic sensory neurons, GABAergic motor neurons, and DOPA. But we can also work with primary cells, which we validated, for example, on keratinocytes and endothelial cells. So as this device is a very good base for any studies and any applications then using the either commercially available cells, or could be also customized or tailored cells that you would have developed in-house. Now we have talked about the technology, then we will introduce to you basically some example of readouts that you can basically get and obtain with our technology. As we said earlier, the substrate is transparent. That means that it allows any type of optical characterization using immunofluorescence or any kind of optical imaging derivatives. The other thing that we can do is, because we have the ability to pipette in and pipette out during the cell culture processes, we have the ability to, for example, to analyze supernatants through ELISA technology and others. And we can do also, if people want, we can do some mass spectrometry analysis in addition of the traditional live staining possibilities. The other thing that we will discuss in a few minutes is the ability to also, for us to measure the electrical activities of the neurons using electrophysiology. Before that, we would like to show you what it means a high throughput in terms of capabilities of a device. So we have this unique capability, and so we can use any kind of automates or liquid handling in the laboratory. So it's very speedy presentation where you can see that you can use automates to do the preparation of the chips. So it's a really high throughput devices. Okay, so let me give the talk back to now our expert in electrophysiology, Dr. Hélène Gauthier, that will talk to you about electrophysiology technology.
So, hi, I'm Hélène Gauthier, I'm a neuroscientist, and my specialty is electrophysiology. And I would like to explain to you how our device, combined with electrophysiology, can allow the recording of electrical activity of the neurons with multi-electrode array technology. So the 2D MEA technology is an array of planar electrodes, and each electrode records the extracellular action potential that we call spike. And here you have an example on the right of the screen. The MEA answers the following question. It will tell you if a neuron is active, and you will be able to differentiate between a healthy neuronal population or a disease population by looking at what we call the activity matrix, thanks to the spike detection. So we have, as a software that we're using is detecting spike, so each activity of the neurons, and the mean firing rate is one of the main metrics. Second question that we can answer is, are neurons forming synapses, and are those synapses altered in a condition, which can be with toxicity, or it can be also a genetic modification. So by looking at metrics from burst and the metric measurement like the synchrony index, we can access synapse health, synapse function. And last, we can also assess with this technology the network formation of neurons, which will be very important for CNS culture. And this activity, those neurons can be altered in different conditions. So we will look at metric, what we call network burst metric, to have this information. And this is an example of how the usage of the MEA with NETRI chip and the advantage of this compartmentalization. So Thierry mentioned before compartmentalized microenvironment, which means that you can really have recordings from the soma. And like, for example, if we think about DRG, look at spatial ectopic activity on the soma, you can look at neurites specifically, or new axons, thanks to the microchannel amplification. So on the top, you will, these electrodes here, the signal is from the microchannel, and you can see that it's slightly higher detection than in the channel itself. As I said, we can segregate soma from the synapse and from axonal activity. You can also measure infer conduction speed or directionality with a dwelling shift. So here I show an example. Either the cell were seated in the channel 3 and growing towards channel 2 and 1, or they were sitting in channel 1 and we will measure and growing towards channel 3, and we will measure the signal between in the microchannel 2 to 3. And when we look at the activity of the electrodes along the microchannel, we can see the signal propagation that match the way to seed the cells. So you can look at directionality. This way you can recreate anatomical network connection and make sure that they go in the proper way and not backward. And you can really study drug effect on a specific population or specific area of your cell, and while keeping physiological connections. So to summarize what I tell you and to show you a little bit into images, so this is our devices and how they are combined with the Axion MEA technology. And so both technology are in the same plate, and you will put out the device in the Maestro machine. And those are examples of the different culture you can do and the type of result and matrix you obtain with the first high throughput compartmentalized MEA that we supply. So to summarize this part, thanks to the MEA and the Maestro system by Axion Biosystem, we've been able to add the electrophysiology readout, to add real-time measurements, functional measurements of the neurons.
So I'm just going to go ahead with the follow-up. So I'm Hugo Cochet. I'm the head of partnerships here at NETRI. Thank you very much, Helen and Thierry, for the introduction. I'm just going to get a little bit more into detail with the solutions that NETRI provides. And for that, we're going to start with what we call the neurofluidics line, which is our basic line for the products that we sell. As you can see, there exists two versions, one called the light version, the other the full version. And the difference is that with the light, you're going to have 8 chips, so 8 data points. And with the full version, you're going to have 16 chips. So just to be very clear, please keep in mind that each neobento is composed of 4 quarter bentos, and each quarter bento is composed of 4 chips, so 4 data points. As you can see, with the light version, the 2 quarter bentos from the bottom have been replaced with what we call plugs. Those plugs are designed so that you are able to put water into them in order to avoid dehydration during the culture phase of your cells. Now, we have the same devices, but with the electrophysiology aspect that Helen just presented to you. And in this case, we're going to have the products called Maestro Edge version and Maestro Pro version. The differences are the same as above. You have one with 8 chips, so 8 data points, and the Maestro Pro has 16 chips, so 16 data points. Please be advised that those devices only work with the Maestro machine from Axion Biosystems, and Maestro Edge is going to work only with the machine called Maestro Edge, and also with the Maestro Pro, whereas the Maestro Pro version will only work with the Maestro Pro. Now, those are the devices that we sell as a product, as a finished product that you can order and that you can receive by passing an order through Coma Bio. And we also have the capacity to sell softwares with the Neurofluidics Digital, which gives you the capacity to analyze and get more in-depth data of everything you're getting, every readout you're going to get from our devices. Also, we have the capacity to sell R&D services, as well as screening services and code development. And this you can see here, where we have what we call the pyramid of our services. First one at the bottom is the NETRI devices, so the ones that I have presented above, and are the ones that you can order and receive directly at your laboratory. And we also have the capacity to realize custom models and assays in-house, so in our laboratories, when we are working with you and for you. Now, finally, we also have the capacity to work together with you in order to develop either new models or develop new drug developments.
Thank you, Hugo. And now I will show you some use case and application of our devices, So NETRI started with a focus on neuron, either in the central nervous system and peripheral nervous system. So briefly, the central nervous system is composed of the brain and the spinal cord that are inside our bone structure, and the peripheral nervous system will be also nervous components that are not into a bone structure. You can see in this slide really the compartmentalization, and I will show you example either of motor neurons and how we can reproduce energy and this separation of the cell body of the motor neurons that are in the spinal cord from the neuromuscular junction at the periphery. I'll show you an example where we use sensory neurons. So here you can really see, and Thierry mentioned it, so the skin that we can collect keratinocyte and other skin cells and have the endings of the sensory neurons. Those sensory neurons have really long axons, and their cell body are in the dorsal root ganglia, and then they connect their second axons to dorsal horn neurons, and the signal will either go through a short loop to interneurons and ventral horn motor neurons, or will reach the brain for area of consciousness. And here in the brain, you will have different areas, and we can also, with our technology, try to reproduce those brain areas. And I will show you an example. So first use case is regarding pain and injury application. So you can use our platform to reproduce injury. Here's the example of the dual link shift that is composed of 3 channels to isolate the cell population, but mostly the cell as each compartment of the cells. So you will use a dual link shift, and we tested with either sensory neurons, or you can use motor neurons depending of your interest. They will grow their axon towards channel 3. When they're grown enough, you can use the channel 2 to create an injury, so an axotomy. Either we recommend detergent application, but we have users that do prefer to use vacuum or mechanical way to cut the axons. And the channel 2 here is also used to apply different drugs that will modulate axonal regrowth. And the channel, as the microchannels that are connecting channel 2 and 3 are of suitable lengths to follow neurite growth kinetics. So here is an example of what we did with culture of eye cell motor neuron from Fujifilm. We let them grow at day 7, so we do have enough neurites in the microchannel 2 to 3, and perform the axotomy only in the channel 2. And we have a robust protocol that doesn't affect viability. And then we added different drugs in the channel 2 and look 3 days later at the regrowth by using a live dye calcium. So here you can see a picture of nocodazole. It's a drug that destabilizes microtubule, and you have retraction of the neurites. In the vehicle, you can see growth of the neurite in the macrochannel, and they start to reach the channel 2. Channel 2, and they regrow in channel 2, and there is a few starting in channels 2, microchannel 2 to 3. If we look at staurosporine, which is our positive control, you can see that the channel 2 is already full. There is already a lot of neurite into, and several of the microchannels are also filled. So to summarize here, so I said that nocodazole destabilizes microtubule, and we could see that at 10 micromolar, it's cytotoxic, and by measuring the viability. At 1 micromolar, however, it reduces neurite outgrowth. We measured by a semi-automated way the neurite growing, regrowing in the microchannel. So we could measure the length, sum of length, or the maximum length, also the number of axons and the mean length. And we could really see that the nocodazole really decreases all those parameters and can be used as internal control for neurodegeneration in your assay. The staurosporine is a wide spectrum kinase inhibitor known to enhance neurite outgrowth. And in our condition at 50 nanomolar, could see the increase in neurite outgrowth, and we can show, we confirm that it can be used as a neurotrophic control, so you can really have 2 controls to compare your drug with. Now, a second example is how to use our platform to study toxicity. And the example here is chemotherapy-induced peripheral neuropathy. So we use, in the data, we'll show you the Dualink Ultra, where the channels are connected by macrochannels, macro grooves that will allow an enhanced axonal projection, and it will also allow you to kind of organize the distal axons. The advantage of this architecture is to be able to apply drugs depending on their mode of action. So here I will show you the example of paclitaxel, which is a lot used as a chemotherapy agent when it's applied on mature neurons. So it's been applied only on the channel 3, like the vehicle, on the mature culture. So the neurons are 20 days before we apply 3 doses of paclitaxel. And after 72 hours of exposure, the cells were fixed and stained for beta-3 tubulin, used as a marker for neuronal health. So we measure the surface covered by the axons and the axonal debris, the blebs that are created when neurons are degenerating. What we could see is that the surface covered by the axon is reduced at the highest dose, as you can see in the graph on the left. The number of axonal debris is increased at the highest dose, and we can really see this dose dependence of the degeneration of the axon, and really recreate this dying back that is characteristic of paclitaxel toxicity. We also perform the same with oxaliplatin, and I encourage you to look at our application note, chemotherapy-induced peripheral neuropathy on chip model, utilizing the strengths of compartmentalization. So the next step is to combine and to use our MEA platform and these benefits of compartmentalization to record the electrical impact of each chemotherapy agent on the sensory neurons, and being able to record from each cell compartment, so the effect on the soma, on the neura, on the axons, and build digital signature of each compound. Now I will show you an example of neurodegenerative disorders and how you can use our technology to model them. So I choose to show you the development of our platform for Alzheimer's disease modeling, which was done in with a partner, EtapLab. So we developed this model using dual-link and also the dual-link shift. And our expertise and development was really to put in place the best culture condition for the glutamatergic neurons and GABAergic neurons, and ensure that they were developing properly in our devices. And our partner did develop the injury model using their A-beta oligomer and their technology. So here is the data from the development of the glutamatergic neuron culture and the GABAergic neuron culture in our devices. So we check with the expression of specific markers, so the reduction of pluripotency markers at day 21 and the increase of maturation, of marker of maturation like beta-3 tubulin, or specific one of each population, like the V-glut that is mainly in the compartment where we have glutamatergic neurons, and the GABA that is mainly in the compartment where we have GABAergic neurons, showing a really good isolation of each cell population. And we look at the development of the electrical activity. So here you have an example of the raster plot, as a compartmentalized raster plot. So here, this bottom light blue will represent the this channel 1 and all the electrodes in the gluta compartment. In the yellow and the red will be the microchannel compartment, and here it will be the channel 3 compartment electrodes. And you can, this is across time. So at day 7, we don't have much activity, which is perfectly expected, and we can see development over time of a good electrical activity in each compartment. In parallel, so EtapLab develops the injury by testing their oligomer on population of rodent cortical neurons, also to show the translationality of our devices. You can use both, either human cells and rodent cells, and make the parallel also with in vivo or clinical data. Here they show that there is no effect of the oligomer on the cell count, so on the soma compartment, but they could see a local axonal degeneration in a dose-dependent manner. Last, I will show you an example from one of our users, which is CHU Saint-Etienne, an hospital in France. They use our chips to develop a platform to assess neuroimmunological communication involved in IBD. What they did is they used a dual-link chip, so two cell compartments. They put glutamatergic, iPSC glutamatergic neurons in the channel 1 and let them grow extension towards the channel 3. And they had stimulated immune cells in the different compartments to look at the modification of electrical activity and influence of one compartment to the other. I will only show you here immunostaining and how the culture look like. And I will invite you to look into more details in the publication for the sake of time today. But what we could, I will briefly say that we could observe secretion of ELCs that was linked with the addition of the immune cells and also change in the electrical activity of the glutamatergic with the addition of the cells. So to summarize, although NETRI started with application on neurological disorder and PNS disorder, we are now developing application for the gut and also for the skin. The application are really broad, and you can also consider many other organs. NETRI offer high throughput and interoperable solution, and really I really insist on the benefits of the compartmentalization, either to have different cell type or to isolate each cell compartment, that the culture is complex and you don't need to have tubes outside the incubator, and is compatible with most lab equipment. Our vision is really that we believe that all organs could be addressed, and by using the right combination of neurons and organ cell, would really be able to have a robust organ modeling, and really by combining the specific readout in a dynamic and non-destructive response. And all of this is because we are building a digital library, so extracting with the electrophysiology, the digital signature, either of healthy or an injury, in order to be able to test efficacy or toxicity relative to the reference compounds. And I will thank you very much for this, for the part of the presentation.